Good morning, everyone. Today, my teammates MD Jihad Hasan, Simur Rahman Fahim, Souvik Mitra, and Junaid Al Habib Siam, Tanvir Hossain, and I are excited to present our topic molecular phylogeny, understanding evolutionary relationships through DNA and protein sequence. We will be exploring how genetic evidence allows us to map the tree of life with incredible precision. In the slide, what is molecular phylogeny? We have to know what is molecular phylogeny. It is a sophisticated branch of analysis that uses genetic and molecular differences, specifically from DNA and protein sequences, to understand how organisms are related. It gives us deep insights into the historical patterns of the life on Earth. In slide 3, we have to focus on inherited variations that accumulated over evolutionary time. By comparing these variations across species, we can determine evolutionary pathways. This process results in phylogenetic trees, which are visual maps of biological history. How do we track these changes? We use molecule markers, think of them as genetic fingerprints. This provides high resolution data for direct genetic comparison. These are great for looking at border evolutionary distances because it's inherited maternally and evolves quickly. It perfects for studying recent divergences. This is universally present and helps us to see very deep ancient relationships. Thank you. The rest of the slide will cover by our next teammates.
Hello everyone, I am Mohammad Junaid Al Habib Siam. ID 253-15769. Today I will discuss the workflow of molecular phylogeny. Understanding evolutionary relationships is not a guessing game. It follows a clear scientific purpose. This workflow has 6 main steps. First is sample acquisition, where we collect biological samples for organisms to create DNA. Second, genetic sequencing, is in which the expect DNA sequence is identified. Third, sequence alignment, where sequences are compared to find similarities and differences. Next is phylogenetic inference, where statistical methods are used to determine evolutionary relationships. Then coloredogram construction, which visually represents those relationships in a phylogenetic tree. Finally, biological interpretation, where we analyze the tree to understand evolution, specification, and divergence. Moving into the next slide, this slide shows the main DNA sequencing technologies used to read genetic information. First, Sanger sequencing, which is a traditional method known for high accuracy on small samples. Next generation sequencing, NGS, which can sequence millions of DNA fragments at the same time. Depending on the study, those methods are used to whole genome sequencing or targeted gene sequencing to analyze specific genes. Moving into the next slide, this slide shows 3 main computational methods to build evolutionary trees. First, distance-based interference, which measures overall genetic differences and works fast for large datasets. Second, character-based analysis, which looks at specific mutations to find the tree with the fewest changes. Finally, probabilistic frameworks, like maximum likelihood, use statistical methods for the most likely evolutionary paths but need more computing power. Thank you, everyone. My team members will continue the rest.
Good afternoon, everyone. This is Jihad Hasan, to provide 317. Mass life topic is deconstructed hydrogenicity. To understanding the history of life, we use hydrogenicity, the visual blueprint of evolution. At the basic base, we have the root responsibility, the last communication ancestors from their basic and needs map out specification event and leading to leading who is identity modern specific and their shared line is. Next slide please. Slide 9. 8. The molecular flow. But how do we put a data on their connected? That's where the molecular flow comes in. This is concept really on the face that genetics by cleaving their genetic things again social record. We can we can issue the same recover on time. Calculator explicitly when two special different appear on their evolution timeline. Slide 10. Advanced application. Finally, why does this matter today? Molecular analysis is a vital tool for connectivity. In research evolution, we use it to track viral transmissions like SARS-CoV-2. In conversation biology, it helps us protect genetics diversity in species, for taking humanity evolution to facilitate new life in microbiology. We stop it for success the hope that one's life energy to pay attention to traditional use case, but how to do we put data on the connection to slide. Okay, thank you. Please next my team member is coming.
Hello, I am SM Tanvir Hossain, ID 253157775. Now I will briefly cover the challenges and future of molecular phylogeny. Challenges and limitations. Molecular phylogeny faces several hurdles. Biologically incomplete lineage sorting and horizontal gene transfer can scramble genetic signals, making trees hard to reconstruct. We also deal with convergent evolution, where unrelated species look similar, and a sparsity of fossils for dating. Technically, the field is restricted by computational intensiveness and a heavy model dependency, where the wrong assumptions lead to invalid results. Next slide, please. Next slide, please. Future directions. The future, however, is bright. Long read sequencing and ancient DNA are allowing us to see deeper into the past with higher accuracy. We are now using metagenomics to study unculturable microbes and machine learning to process vast datasets efficiently. Ultimately, by integrating phylogenomics, combining genetic, ecological, and physical data, we are building the most complete map of life's history yet. Now, I would like to invite Fahim for next topic discussion. Thank you.
Thanks, Tanvir. I'm Fahim, ID 253-15741. I'll discuss about further topics. Key insights in molecular phylogenetics. This slide summarizes the major insights of molecular phylogenetics. Molecular phylogeny reveals evolutionary relationships by comparing DNA and protein sequences, helping us understand how species are genetically connected. It uses an advanced methodological arsenal combining modern sequencing technologies with powerful computational tools to build accurate phylogenetic trees. These trees help chart historical divergence, showing when species split from common ancestors. Using molecular clocks calibrated with fossil records, scientists can also estimate the timing of key evolutionary events. Overall, molecular phylogeny has a strong traditional impact, supporting medical research, conservation efforts, and studies of human evolution. Next slide, please. Now, foundational principles of molecular phylogeny. This slide explains the principle behind molecular phylogeny. The foundation is genetic homology, where genes or proteins inherited from a common ancestor are compared to detect evolutionary divergence. Phylogenetic interference methods such as maximum likelihood and Bayesian analysis are used to identify the most probable evolutionary trees. Molecular clocks estimate divergence times, while data robustness achieved by using multiple genes and statistical validation ensures reliable results. In conclusion, molecular phylogenetics combines genetic data, advanced computational methods, and evolutionary theory to reconstruct the history of life. Its wide applications and continuous innovation make it an essential tool in modern biological research. Thank you.